Hello everyone, and welcome to today's live broadcast, Development of Tools Enabling Tunable in Situ Delivery of Therapeutics Using Probiotics, presented by Nathan Crook. He is a postdoctoral researcher in the Department of Pathology and Immunology at Washington University School of Medicine. I am Marjorie Torres of Lab Roots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Lab Roots. Lab Roots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Nathan Crook, and I will now turn the presentation over to him.
Thank you very much, Marjorie, for the kind introduction. I want to thank you all for viewing this webinar. I'm really honored to have been chosen to present to you guys today. And what I'll be talking to you about is some of my postdoctoral work, engineering probiotics to enable them to eventually deliver therapeutics in a tunable in vivo way. So my upbringing was in chemical engineering, and some of you might want to know why a chemical engineer would choose to engineer microbial communities. And my answer to that is that microbial communities are actually one of the most sort of beautiful, intricate structures that I have the privilege of seeing. What you're seeing on this slide are the microbial communities associated with anaerobic sludge digestion. This is the process by which microbes actually ferment or organic liquid waste into chemicals that can be more easily processed. And in this image is one granule, which is composed of different microbes, and each of these microbes is stained a different color. The aerobic microbes are colored green, and the anaerobes are colored in red. And it was very interesting to me to see how these microbes co-associate into these intricate 3D structures. Now, these microbial communities exist not only in our industrial settings, but also on our body. What you're seeing on these pictures is an image of dental plaque. And it's really exciting because what the researchers did in this paper was they took people who hadn't brushed their teeth for a couple of days, and they scraped the material lining their teeth and imaged that on a microscope. And they stained each species of bacteria a different color. And what was extraordinary to me was the fact that these microbial communities actually associate into sort of the green layer on the outside and the purple layer on the inside. And these actually represent different microbial species that do different metabolic functions. The microbes it's colored in green are aerobic, which makes sense because they're on the outside of this plaque. And the microbes in purple are the anaerobes, and they are inside because there's no oxygen in the environment. Not only do they form this unique porcupine structure, they also have, if you look on panel F, you'll see honeycomb structures along the filaments made by these probacteria. So the fact that these microbes are able to self-associate into these intricate nanostructures kind of really motivated me to get into this field. Now, these exist not only in our teeth, but also in our gut. And so if we zoom in on what a bacteria sees when it's in our gut, you'll see an image where different players within the gut are seeing different colors. So in blue here are this is a mouse gut, this is mouse epithelial cells. In green is the mucus that these epithelial cells are secreting, and this mucus serves three different functions. It not only provides food to the bacteria, and it also protects the host from bacterial, potential pathogenic bacteria, but also lubricates the flow of material as it passes along the gut. So the mucus is actually very important to bacterial metabolism, as we'll see in a few slides. If we sort of take a broader view of the gut and we stain different types of bacteria based on their sort of taxonomic identification, their species, you see that there are many different types of bacteria that exist in the gut. And here they're staining the Bacteroidetes and the Firmicutes within the gut. Certain types of bacteria associate toward the lumen or the interior of the gut, and those are fermenting, undigested material, whereas other bacteria associate with the mucus and are actually fermenting the mucus as a food source. As sort of a fun exercise, what I did is I took mouse fecal material and resuspended it in phosphate buffered saline to see whether these bacteria are actually metabolically active, and if I could see that on a microscope. And what you're seeing on this movie is the image that I saw on my microscope. You see a lot of bacteria swimming around in fecal material. Much of this might be due to Brownian motion, but you also see bacteria that appear to be moving in a more directed fashion. They're kind of zipping around this image very quickly, and they're clearly metabolically active within fecal material. And I just thought it was amazing to see the diversity of shapes, structures, and behaviors exhibited by these bacteria within the gut. So not only are these bacteria microbial communities beautiful, but they also serve a very important biological function. So if we take a look at the human body itself, the human body contains a diverse array of genes, 23,000 gene products, myriad cells, and a very large biomass. Contained within the large intestine, though, is this diverse microbial community that is similarly amazing. It has much orders of magnitude more genes encoded within it than the human body does. It has about the same number of cells as the human body, but it also exists in a biomass that is not insignificant. It could be used for doing metabolic and biochemical conversions. What it naturally does is it takes that undigested material from the gut and provides about 10% of our daily caloric intake through digestion of this fiber. It synthesizes vitamins for us from that undigested material, in particular vitamins B and vitamins K. Just by being in this nutrient-rich environment and not doing anything particularly pathogenic, it also resists pathogen colonization. Excitingly, the membership of this microbiome is dynamic. It can change over daily and almost in yearly timescales as well. In addition to its natural variation, it can also be perturbed in a more directed way through probiotics and fecal microbiota transfers. So this dynamicness is part of why I'm interested in it, because you can potentially engineer this microbial community. So what does this microbial community look like in healthy individuals? In healthy individuals, if we look at just the bacteria within the gut, you see a large diversity of memberships. So on the sort of side of this graph here, what you're seeing are the different phyla that are the that make up this microbial community. You see Bacteroidetes, you see Firmicutes, you see Proteobacteria in varying ratios within the gut. All of the microbial species abundances add up to 1 in this graph, so this is a relative abundance measure. The Bacteroidetes are the types of bacteria that are commonly associated with fermentation of long-chain polysaccharides within the gut. And the Proteobacteria are the more famous members of which include E. coli, for example. And those exist as well within healthy individuals. So if we go to the next bit, you can also see fungi within the gut as well. So fungi exist in healthy individuals' guts. These exist in commonly known genera, including Candida. Now, Candida is commonly associated in the medical literature with yeast infections, but there's also non-pathogenic versions of Candida which exist in the gut as well, or at least they don't. There are pathogens that are not contributing to an infection. There's also Saccharomyces involved in the gut, and you might know this genus of fungi as the one responsible for making our bread and brewing our beer, and this also exists in our gut as well. If we take a look at the gastrointestinal tract in relation to other body sites, so other body sites also have these diverse microbial communities. You see the gastrointestinal tract localizes toward a particular region of this non-dimensional space. So this graph is what's known as a principal component analysis plot, and it takes all of the multidimensional data for the different microbial species abundances and condenses it down to one point in the graph. Now, points that are close together on this graph indicate very similar species distributions, whereas ones that are further apart indicate microbial species distributions that are very different. So if you look at this graph in sort of a qualitative way, you'll see the gastrointestinal tract is very different from our oral microbiome or even our skin microbiome. Interestingly, the urogenital tract exists kind of in the middle between the gastrointestinal tract and the skin. If we look at this microbiome in correlation to disease, you see that individuals with the disease listed here have been shown to have microbial species abundances that are very, very different from what you see in a healthy individual. In particular, obesity, autism, and colon cancer are very hot topic issues that people have shown that if you not only is this microbial community different, this microbiome different, but also if you give a diseased microbiome to a healthy, in general, mouse, you see you'll see the showing of disease-like symptoms. So the fact that the microbiome is so important to health and so important to disease indicates that the gut microbiome is actually kind of like an organ in the human body. And the upshot of this is that this is the only organ within the human body that can be engineered without surgery, because you can do these sort of things like give probiotics or fecal microbiome transfers. So what type of things can we do to the gut to enable it to be engineered? So one of these things is a probiotic. These are live microorganisms that improve gut function. These are defined species. So I'm showing you 3 examples of different types of probiotics which people will take. One of them is E. coli Nissl, shown on the left here. It's a large intestinal prokaryote, so it's exists in the large intestine. It's effective for treatment of inflammatory bowel disease, diarrhea, and constipation. It's actually marketed in Europe as Mutaflor, and it's one of the few sort of probiotics which has been clinically shown to have improved the ability to treat these sort of irritable bowel disease infections. There's also Lactobacillus rhamnosus. This is a bacteria that lives in the small intestine. It's also effective for a variety of conditions. And I want to draw your attention to Saccharomyces boulardii. This is a eukaryote that exists in the large intestine, and it's effective against many different types of pathogens. It actually binds to and inhibits the toxicity associated with C. diff, H. pylori, and Salmonella typhi. So not only are prokaryotes very useful probiotics, but the sort of neglected eukaryotes are also effective probiotics as well. Now, unfortunately, these probiotics are sort of natural organisms. They have a certain amount of limitations to them if you want to use them for therapies. One of them is a low residence time within the gut. What I'm showing here is a graph of E. coli Nissl abundance over time in individuals who took this probiotic. So as you can see, as the weeks go on in this study, the percentage of individuals who were positive for E. coli Nissl steadily dropped. And so this is a problem because E. coli Nissl is commonly taken for treatment of inflammatory bowel disease, and which is a chronic condition. So if you have a chronic condition, you want a chronic treatment, and probiotics do not satisfy that criteria. The other problem is you have a limited amount of expression control with these probiotics. So if you want to deliver a particular therapeutic dose of a gene product to the gut of an individual, and in this case I'm showing a mouse for experimental simplicity, you only really have a couple of options. You can either make it very lowly expressed, or you can sort of ramp up the expression to some high level that you're not really sure what it is. So you have a limited ability to tune this potentially toxic gene product. So it might have a therapeutic window that you don't want to get out of. And so you need very detailed expression control in this case. So in response to these limitations, in my postdoc, I've been developing several transformative technologies for the gut. One is to improve the rate at which probiotics can colonize, and the other is improving the resolution with which we can tune a gene product. So in case this wasn't clear on the previous graph, the y-axis shows sort of a phenotypic response in relation to the dose of a particular gene product. And so hopefully there would be some optimum that coincides with the therapeutic window of that drug that you want to tune. These two things might seem quite distinct, but in reality, they share the common theme of high throughput screen in them to enable their ends. So before I get into the colonization enhancement aim of my work, I want to first thank the people I worked with. As you see on the top view, from left to right, Terrence Crofts, myself, Sherry Sun, Ara Ferriero, and Drew Gasparini were all very instrumental to the continued development of this work. On the bottom, you see Mitch Pazewski, Molly Gibson, Bean Wong, and of course my advisor, Gautam Dantas, were continue to be instrumental in both the conception and current development of this work. So when we envisioned improving gut colonization, we had a lot of ideas about what might impede a new organism from living within the gut. There's of course the physical flow of material within the gut that pushes the bacterium from beginning to end. There's there's phage or viruses within the gut that can predate on these newcomers, as well as the immune system of the host itself that can interfere with the growth of a newcomer. Finally, there's a existing metabolic network that that the existing bacteria play a part in, which may exclude a newcomer from participating in. So this is all very abstract, but if you want to sort of make the analogy to moving to a new place, bacteria need to learn the cuisine of a new location when they go into a new habitat. They need to have there's actually a bacterial language barrier that they need to overcome in terms of interspecies signaling. Niche competition is like finding a job. They need to know, or they need to be able to convert metabolic material from one form to another in a downhill fashion in order to make their living in the gut. And there's also local diseases that they need to gain resistance to. So there's a lot of barriers to living in a new place for a bacterium, and without knowing which one was the sort of rate limiting step, we opted for a non-rational approach to engineering this strain. Now, what does that mean? In order to figure out sort of how we engineer bacteria via non-rational approach, we have to know how we engineer bacteria in general. So as you guys are all probably well aware, DNA is sort of the recipe book of the cell. The gene that encodes a particular gene product is transcribed into messenger RNA, which is transcribed into a protein, which performs some function within the cell. I like to make the analogy to a cookbook where a gene itself is like the cookbook, where it's never really taken to a kitchen, but contains all the necessary instructions for the thing you're trying to make. You'll transcribe that cookbook onto a sticky note and take that into your kitchen to make your dish. And your dish is something that you would like to eat. So the question that we had to answer for ourselves is, how can we find new genes that do interesting things? And this is we wanted to make this as a high throughput and non-biased approach as possible. So if we continue with the analogy of finding new recipes, if we want to find a new recipe that does something that we want, how would we do it? By brute force. Well, the way we would potentially do it is take all the recipe books that we had access to and take random pages from each one. So just randomly tear pages out. We have to give these random pages to the best cook that we know, the cook to sort of make these into an actual dish. And the best cook I know is my grandma. So let's say we deliver these recipes to my grandma and have her cook what's encoded within them. Now, some of these recipes are going to give us the food that we want. In this case, it could be the lasagna that we want. But it could be other foods that maybe we weren't interested in, or it could be nothing at all, in this case an empty plate. Now, if we do this enough times, we're going to get lasagna more than once and figure out sort of what constitutes a lasagna recipe and learn from that. So in a similar sort of way, we're going to search gut bacteria to identify genes enabling E. coli and to colonize the large intestine. So instead of cookbooks, in this case, we have bacterial genomes. And these bacterial genomes are contained within fecal material from healthy human individuals. What we did is we took these fecal samples and extracted the DNA that they contain. These are our recipes. And we sheared them to 2 to 5 kb chunks. Now, we did this to sort of narrow our search down to pathways or single genes that would allow us to improve gut colonization. We took this DNA and gave it to our cook, or our, in this case, it's our probiotic E. coli that will transcribe and translate the DNA encoded within these shared products. And the way we identify whether these gene products encode improved colonization capacity is we deliver this probiotic library into mice. Now these mice will shed the delivered E. coli nissle over time. The poor colonizers will be shed quite early, and so in early time points, mouse fecal material will contain pretty much everybody. However, as time goes on, the good colonizers will be sort of latched on into the gut and be producing progeny, and those will be enriched in the fecal material of these mice at later time points. So by amplifying the DNA that's contained within this fecal material that's specific to E. coli, we can sequence it and figure out what was encoded within the fragments that stuck around, and we can learn from that. So if that was a bit too technical for you, basically what we're doing is taking 10 to the 7th genes or gene products from gut metagenomes and narrowing them down to 1 to 10 hits that are good colonizing factors. The exciting part of this approach is the fact that you can take the same basic technique and make it sort of reductionist or learn different things based on how you structure it. So in this case, what we can do is a reductionist approach where we can take the simplest possible way we can think of for gut colonization or the most complex possible way we can think of for colonization. So what do I mean by that? The first experiment we actually did was in germ-free mice. These mice have no native gut flora besides E. coli, of course, that we deliver to them. And it really models just the host E. coli interaction. So what is necessary for colonization in that context? And we can add complexity to this in sort of an iterative fashion. So we can give it different diets, for example, a human diet, or we can add different members of its gut. In terms of these conventional mice, they actually have a gut microbiome that contains other gut bacteria as well. So we can add complexity and learn what was necessary to colonize at each step to figure out what's necessary to colonize. So before I get into the actual results, I did want to touch on a little bit the fact that we did our experiments in germ-free mice, and this is sort of it's a technique that's been around for a while that may not be familiar to everybody. And what it does is it takes mice that are born germ-free and it houses them in these sort of plastic bubbles. These are self-contained plastic bubbles. They have their own sort of sterilized air supply that comes in, and air is sterilized when it comes out. Everything that's given to these mice in terms of food, water, bedding, etc., is delivered via a sterilized cylinder and given to the mice. So everything is sterilized when it comes in, sterilized when it comes out, and we bleach this plastic cylinder to make sure that no bacteria are currently residing on it. And we actually never touch the animals ourselves. We use these gloves to make sure that these mice exist in an environment that is completely, of known bacterial composition, or metabotic in this case. And this is a very powerful approach because what you can do is do experiments on germ-free animals, so animals without any microbes living in or on them. You can do experiments with animals that have a known bacterial composition. So you can take these individual microbial species and culture them in the lab, and then you can have a handful of a sort of synthetic microbiome that you can do experiments on. And that's sort of the bottom-up approach, and the top-down approach to that would be to use human fecal material from individuals who are diseased or healthy and deliver that to those mice and see their effects on mouse phenotype. You can do these with mice, and people have done, obviously the complexity increases as you increase the size of the animal, but it's a very powerful approach to answer hypothesis-driven questions on the gut microbiome. So let's get into the actual results. We did our first experiment in completely germ-free animals that were eating a mouse diet. We had 6 animals in this case. On day 0, we gavaged our library. This was a 10 to the 7th member E. coli nissle library where every member of this library had a different gene product from a gut metagenome. And we took fecal pellets over time, over the course of 3 weeks. At the end of 3 weeks, we collected cecal contents, and this is the different colored material here. What we saw was really exciting. On all of our 6 mice, we saw enrichment of a handful of gene products to near 100% abundance. So on the y-axis of all these graphs is the stacked abundance, or the cumulative abundance of the DNA fragments that I'm showing. So 100% means that all E. coli contained within that mouse have the fragments that are listed on the graph. And time is on the x-axis. And over all of our 6 mice, we had, excitingly, the same 5 DNA fragments enriched in these mice. So all these fragments are colored consistently. So the blue fragment is the exact same DNA sequence in all 6 mice. And from our 10 to the 7th member library, every single one of our mice that we chose was able to enrich for that particular gene product, indicating that this blue and the orange gene products are very important for gut colonization. We also saw a lot of other gene products which were very important. So before I get into the actual sequence of those genes, what I'm going to show you is our next experiment where we took germ-free mice again and housed them with different human-relevant diets. So I'm showing another graph here, which is similar to that shown above, where we have 15 germ-free mice, and they ate a human diet. Now, this human diet is sort of loosely defined, where it's actually a high-carb, high-fat diet. So it's more mimicking a Western-type diet than anything else. And we wanted to do this because we were interested in, probiotics that, the differences between a mouse diet and a human diet. We also added one of several prebiotics. In this case, we used benefiber, inulin, or just, our control was no prebiotic. And prebiotics are long-chain polysaccharides that actually pass through the gut undigested and make it into the large intestine. And they serve as food sources for the bacteria. So we were envisioning that these prebiotics would actually select for probiotics that were dependent upon them for growth. In that case, the combination of a prebiotic and a probiotic is called a symbiotic. What we saw was also very, very interesting. In this case, like before, in all of our replicate mice, we saw the same types of DNA fragments enriched in each one. So in this case, in benefiber, you see the same colors appearing again and again. Those indicate the same DNA sequences are recovered in those mice. You also see that over time, over the course of our 5-week experiment, we were able to sort of capture nearly 100% of the diversity in those large intestines with these fragments. But what's interesting is that between the arms, we got a different set of DNA fragments recovered in each one. So in benefiber versus inulin, for example, we got very different DNA fragments recovered. This indicated to us that maybe these DNA fragments are specializing toward the particular diets that these mice are eating. The other thing we thought was interesting was that there was one fragment that was recovered in all 15 mice. Even though these mice were separately caged, they were able to recover the same purple DNA fragment in each one. And this indicated to us potentially a generalist DNA fragment that was sort of able to colonize regardless of diet. So we were very interested in these sort of generalist fragments that we recovered in this experiment as well as the experiment prior. We then sequenced these fragments and discovered that these fragments actually encode carbohydrate utilization loci. So on the side here, I'm showing the fragment name and the predicted source of that fragment based on a BLAST search. The MD fragments are those fragments that were recovered from our first experiment, and the HD, or human diet, fragment was the generalist that we recovered in our second experiment. On the graph on the scale is the sort of scale, the length scale of these fragments. In light blue is the promoter that is driving expression of these fragments, and every arrow contained in these fragments is a different gene. And these genes are colored based on their predicted annotation in the predicted function based on the hidden Markov model. Excitingly, most of these fragments contain glycosyl-hydrolase family 32s. These are enzymes that are predicted to hydrolyze sucrose into its constituent monomers. Sucrose is actually a disaccharide that E. coli nestle can't actually utilize. These were excitingly also located proximal to predicted transporters. So we were excited to think that maybe sugar transport as well as hydrolysis was something that was limiting E. coli nestle from colonizing. So what we did is we did a couple of directed growth experiments in minimal media with different carbon sources as their single carbon source in order to see which one in particular these guys were allowing consumption of. What we saw as expected was that sucrose was one carbon source that allowed growth that allowed privileged growth of these fragments over a no fragment control. So no fragment control is shown in green, and what you're looking for is growth above the green line. In this case, in sucrose, the green line is here. And you see that all of our DNA fragments except one, were able to grow faster on sucrose than this no fragment control. This is our no fragment control could be thought of as wild type missile. But we also saw other carbon sources like raffinose and also stachyose, which were important for, actually enabled privileged growth of these fragment-containing micelles versus our wild type. And they actually enabled faster growth on these more complex carbon sources than sucrose, which is very simple. We did see one fragment, the one fragment MC4 that grew faster in galactose was the only fragment that could grow faster in galactose, and it could not utilize any of the other 3 carbon sources. So between these 4 carbon sources, we've kind of figured out what these carbon sources are doing. And if you look at the actual structure of these polysaccharides, you see a conserved theme where every one of these has a sucrose head and several glucoses sort of trailing off toward the end. And this makes sense to us because potentially, the annotator was telling us that yes, hydrolysis of this glucose-fructose bond was very important, but maybe these different fragments are adapting toward either more simple or more complex carbon sources. Now, what we don't know is whether these carbon sources are derived from their diet or if they are derived from host, and that more experiments will be necessary to figure that out. So next what we did was we took these fragments and we wanted to figure out whether they would enable colonization of a more complex mouse microbiome. So we took all of our 20 generalist and specialist fragments that we identified in prior experiments, and we cloned them out and transformed them into E. coli micelle in a fresh strain. This would allow us to sort of account for any genome evolution that had occurred in previous experiments and just assay the actual fragment itself in terms of what it would do to colonization. And we compared this to the colonization of an empty vector containing E. coli micelle in 2 separate arms of the mouse experiment. These mice were either containing the fragment library itself or the empty vector wild type micelle. What we did was we took 24 specific pathogen-free mice and we allowed them to first, sort of equilibrate in their habitats, but we also did we had to give them antibiotics 3 days before gavage of our E. coli, and that was because conventional mice actually exhibit high colonization resistance to E. coli micelle. E. coli micelle can't colonize at all in these mice without prior antibiotic knockdown. This actually kills a lot of the resident microflora. So this is definitely a simpler or sort of a less complex case than a conventional mouse, but it's more complex than the germ-free mouse. So it's an interesting sort of stepping stone toward colonization of a conventional mouse microbiome. In the benefiber arm, we saw something quite exciting where the fragment containing E. coli was able to colonize to a higher abundance at the end of our experiment than any of our empty vector containing E. coli strains were. We did this by assaying the intestinal contents at the end of 5 weeks, and we saw that in the proximal small intestine, so the beginning of the small intestine, as well as in the cecum and colon, the E. coli that is contained within the E. coli that contains our fragments was able to colonize much more robustly than the empty vector containing E. coli was. And so we're trying to figure out which ones of these 20 fragments enabled colonization and also why, because that is also a very, very important question. So this is an exciting result that motivates us to continue working on this. So that's the current status of that project. The aim of this was to improve probiotic efficacy by increasing gut persistence, and we used an approach called functional metagenomics to identify colonization factors. We identified that sucrose and galactose utilization were very important for survival, and we also found that these same colonization factors that you can isolate in a germ-free case also translate to a conventional mouse or a specific pathogen-free mouse and allows colonization there as well. So looking forward, I'm excited to use this approach to identify improved colonizers in other body sites. We can envision delivering to other organs, the skin, small intestine, stomach, etc., and identify those bacteria that exist better in those cases as well, as not only for E. coli micelle, but also for more strains that naturally exist in those as well, in those settings as well. So let's move on to the promoter engineering part of this work. I first want to really thank my undergraduate who worked with me on this project. His name is Zevin Kondiot, and he was a fantastic, sort of colleague to bounce ideas off of and really get this project off the ground. So, well, when we thought of this project, we had a particular problem, right? What we would really want to do is deliver a particular therapeutic gene product at different doses and to a mouse to see what effect was most effective for the mouse's health. Now, this gene product could be a recombinant antibody or a particular gene that allows consumption of a novel dietary material. And there is likely a therapeutic window for this gene product which you don't want to go out of, and there's likely an optimal gene expression. So the way you normally would do this, if you had a library of promoters, is you take several different promoters, ones with different known strengths, and put it in front of the coding region of your gene and deliver that into your strain. And that's the experiment you would like to do in order to tell you whether this is working or not. Unfortunately, we don't really have known strength promoters within the gut. We have a lot of promoters that we could test, but we don't really know which ones would work or not, and how well they work in the gut. The reason why is because normally what people will do is we'll take these promoters and you'll put them in vitro. So you take a test tube and you can take 3 test tubes per promoter and you assay its activity via flow cytometry or qPCR to identify how strong each one of your candidate promoters really is. Unfortunately, this works very well for in vitro, but not in vivo. In vivo, if you made the simple analogy of 1 test tube equals 1 mouse, that really gives you too many mice, because in normal cases you want to be testing tens to hundreds of promoters at a time, and you don't want to be using mice in that way. You want to use them in a smarter, more directed way. So we need a high throughput in vivo promoter analysis approaches. So the way in which that I decided to do this was through the following method. What we would do is we would take a promoter library that we wanted to characterize in vivo, and for every promoter that we would test, we'd clone it in front of a reporter gene, and there'd be a unique mapping between that promoter and a promoter-specific barcode at the end of your gene of interest. So you have a promoter, and for every promoter you want to test, you have a unique DNA barcode. And this is a unique DNA sequence that doesn't really interfere with gene expression, but tells you which promoter that came from. What you could then do is generate your library of E. coli strains using each one of those promoters. So every E. coli strain has a different promoter that's driving its reporter gene. Give those to your mouse. So in this case, you don't need 1 mouse per promoter or 3 mice per promoter. You need 3 mice per library of promoters, which is a very substantial reduction in gene expression. And then over time, you can recover fecal contents, small intestinal contents, large intestinal contents, and recover the promoter expression from that. And I'll get into how we actually did that later on, but this is sort of the high-level view of what I'm doing. Is we're taking a promoter-centric approach for mouse experiments to a library-centric approach, and we can measure many promoter constructs in very high throughput. Then what you can do is you can recover DNA and RNA from those organs. So the RNA measures the sort of gene expression abundance, and importantly, the barcode that I inserted after each promoter is going to be present within that RNA. It's also going to be present within the DNA in that construct. The DNA will measure the abundance of that bacteria on its own within the gut. So by calculating the number of each barcode in the RNA and dividing that by the number of each barcode in the DNA, you can get a measure of promoter strength. Now, the advantage of this approach is that it's very high throughput, so you can measure hundreds of promoters in the same experiment, and it's also host and habitat agnostic. So you can take this approach and deliver gene products for eukaryotes, for example, into Saccharomyces boulardii, and you can do it in different habitats like the skin or the root microbiome, etc. So it's a very flexible approach. So the first thing we needed to do was identify which promoters we wanted to characterize. What we did was we used parts previously used for synthetic biology, in particular a 20-member constitutive library developed by Chris Anderson. We also included known inducible promoters by different small molecule inducers like IPTG, arabinose, and hydrotetracycline. We also used an inverter circuit, arabinose and TET-R, which I'll get into in a little bit. But these synthetic promoters, these synthetic constitutive promoters developed by Chris Anderson, span a wide range. This is the reported activities of these promoters, and it would allow us to see whether this mapped over well to the in vivo case or not. We also took known E. coli Nestle promoters, and the way we defined these was through the human gut metatranscriptomic data. It had 8 subjects. We extracted the reads matching E. coli genes, and we took the read count and divided by gene length in order to identify which genes were the most, or in order to characterize the expression level of each gene. We wanted a range, so we took the highest level of expression and the lowest level of expression, and we divided that range into 20 log-spaced bins. Within each bin, we chose the gene which satisfied 2 criteria, one of which had the lowest between subject expression variation, so we wanted it to perform the same way between different subjects, and also was immediately downstream of the promoter, of a promoter region. And we wanted, in terms of bacteria, if you have an operon with many, many genes, we wanted the gene that was most proximal to its promoter to gain the most accurate measure of that promoter's activity. So here are the candidates that we identified. We have some genes that are very high reported expression and some genes that are very, very low expression. So we were excited to figure out, okay, do these actually perform the same way in vivo? What we did is we took these promoters and we cloned them in front of a green fluorescent protein reporter gene, these known Nestle promoters as well as the synthetic promoters, and we characterized their expression via flow cytometry. What we found was that the promoters that we have cloned in span a 100-fold range in E. coli Nestle. Now, it's important to note that this is in vitro growth in LB broth, very, very different from the in vivo case. But they span a 100-fold range, and so this gives us a promising starting point to do further characterizations. Of interest, we did find that our flow cytometry sort of limited our ability to gain access to those really weak promoters. So if we look at the Anderson promoters and we correlate on the y-axis with what we observed versus the x-axis, which is what has been reported, we find a good correlation for the strong promoters, but as the promoters themselves become very, very weak, you see a sort of a tailing off of the correlation, and that could indicate a limited resolution of our flow cytometer at very, very low expression levels. So a more high-resolution approach is necessary there. This also limited our ability to figure out whether our native Nestle promoters were performing as expected, because we have our, on the y-axis, our flow cytometry data, and on the x-axis, the reported data. And when you have a correlation that looks like this, one of your methods is not really that accurate. And I think it's because our flow cytometer is not doing a good job at this range. So we need something more like qPCR or potentially this barcode measurement approach to identify what's going on. We recovered the fact that these inducible promoters function as expected. Actually, the PLAC promoter doesn't work very well in Nestle. Actually, I think what's going on here is that the Nestle is actually being auto-induced by components within the LB broth. So LB broth is not the best broth to use for the PLAC promoter. PBAD is a promoter that's induced by arabinose, and we saw that at different levels of arabinose, shown on the x-axis, we were able to get steadily increased expression on the y-axis. So the PBAD promoter works very well. And PBAD is important because arabinose is generally not digested very well as it passes through the gut, or it passes through the small intestine, so that it can pass largely unimpeded to the large intestine, where you can then induce promoters that you would like to turn on at certain times. We also did an exciting experiment where we used this PBAD TET-R inverter. Now, this has the opposite behavior to the PBAD promoter, where as arabinose increases, the expression actually goes down. And the way this is done is that your PBAD promoter, which is induced by arabinose, actually induces expression of a repressor protein, TET-R. This TET-R protein, as it's turned on, continually represses your reporter gene. In this case, it's our GFP. So as PBAD increases, GFP decreases. And this is known as an inverter or a NOT gate. And both of these sort of turning on and turning off are very, very important for gut behavior. So it's exciting that they worked in a probiotic. What we found is that this choice of strain influences promoter strength. So Nissl activity is shown on the y-axis, and a normal lab strain of E. coli is shown on the x-axis. So just even if it's the exact same media conditions, the exact same growth conditions, we saw that the strain itself actually doesn't influence promoter strength. So this is sort of a motivator to why we would need to do this in vivo measurement case. We found, excitingly, that barcodes, the addition of a barcode does not affect the expression. So this is based on the fact that we need a promoter-specific barcode at the end of your gene of interest. And so the we had the reasonable concern that the addition of a random DNA sequence that is only 6 base pairs and 50% GC at the end of your gene may impact expression to some level. What we saw was that but that wasn't the case, though. We saw largely within experimental error, largely the same expression with and without the barcode. That was very, very good for us, and indicated that the barcode itself would provide allow us to accurately measure promoter strength. So let's get into the actual nitty-gritty details of how we do our experiments. You take your promoter library with a promoter-specific barcode, and I hope you can see this. Well, there's a black mark on my slide, but that's okay. I'll kind of show you what I mean. What's going on is that you have this barcode encoded on either your DNA or your RNA, and after you make your complementary or cDNA, you can then PCR that DNA with primers that hybridize to your intended product. So in this case, it's our reporter gene and our terminator afterward, and they will amplify this barcode. These primers contain not only sample-specific barcodes shown in purple to allow you to multiplex your experiments on the same sequencing lane, but also these things called unique molecular identifiers. These are shown in orange, and these are actually random base pairs that are added during oligosynthesis, so that every single primer molecule actually has a different sequence in that region. And that allows you to uniquely identify these starting molecules in your PCR. So what you do is a small 3 base pair or 3 cycle amplification using these primers, and that basically allows you to uniquely prime one set of primers to one template. And that will uniquely label it with a unique molecular identifier, a sample barcode, and of course your promoter-specific barcode. And then once you have your unique molecular identifiers placed, you can then use Illumina sequencing primers to amplify that region and then sequence it. In this case, what we did is through this whole process, we generated a PCR product that was 227 base pairs in length. And it's very important to gel extract this product, as there are a lot of primer dimers that can form during your PCR, which you want to get rid of. But by counting the number of unique molecular identifiers, you can then get a measure of your promoter strength. So how do we do this computationally? We first take our reads that we got off our sequencer, we trim them to the desired length. We use a program called usearch to merge paired reads together. So we have reads going one direction, one the other direction. They actually overlap at some rate, and you can use a program to correct errors in sequencing that way. We use a custom Python script to count for every promoter and sample barcode combination the number of unique molecular identifiers you observe. And it's important to note that instead of counting the number of instances of each promoter barcode, you're actually counting unique molecular identifiers, because that is able to correct for potential PCR biases during your amplification. And then you divide your RNA abundance by DNA abundance to get promoter activity. The first thing we wanted to do is make sure this type of thing was accurate or not. So we did a standard curve. And what this does is we take each promoter and we give it the same input abundance into our method and run the method through. And we're looking to identify whether your promoters are sort of randomly giving you different measurements out, or they're giving you what you thought you were going to get. And interestingly, what we saw was quite was not as what we expected. What we saw was using this approach that the different barcodes that we were using actually greatly impacted the efficiency of recovery of that barcode computationally or after sequencing. So some of our, what you're seeing on the y-axis is the bias. So this is the amount of barcode or unique molecular identifiers that we observed divided by how many we expected. On the x-axis is the promoter itself, and that promoter is, we're not really measuring the promoter, we're measuring its barcode here. But what we saw was very interesting. No matter what template concentration that we started out with, we did a 2-log dilution in template concentration, we got the same type of bias for every barcode. And we also saw very tight error bars here. These were 3 independent experiments run on different lanes of our sequencer, different preps of our barcodes, which indicated to us that this barcode bias was something intrinsic to this method and not random. It indicated to us that we could actually use this as our standard curve and divide out any our sort of signal that we got from our sequencer by this bias to get the true measure of DNA sequence. Now, the way we tested that was through the use of a sort of an actual curve where we took different mixes of our promoters. So the promoters shown on the y-axis, and we saw what we were able to do is either give a uniform distribution or have some distributions where some were high and some were low, or some were, you know, in the middle, etc. And what we should be able to do is take mix 1 as our standard and recapitulate the expected amounts of mixes 2 through 5. And these are like different types of promoter strengths you might observe in different samples. What we saw was that indeed this approach really does allow you to get at the correct input abundance using your bias you observe in a standard curve. So this standard curve method is something that's a little bit underutilized in the metagenomics area, but it's very important to get accurate results because you can use that curve to really determine what your what the correct absolute abundance of your sample is. So that's the current status of that project. We're currently excited to do it on in actual in vitro and in vivo cases, but what we saw so far is that the strain influences promoter strength. The barcodes themselves do not influence promoter strength, the barcodes used for measurement, but the barcodes do exhibit a consistent PCR bias, and that's something that is very important because a lot of amplicon sequencing is could potentially be affected by PCR bias if this is true. We observe accurate measurement across a 3-log range if we use this barcode bias as a correction. And looking forward, I want to use this method, and I'm excited to use this method in vitro, different media conditions, different oxygenation conditions, and correlate that to what we observe in vivo, different host, diet, and microbiome effects as well. So I want to thank you for your for listening to me talk. I talked about 2 things today. One was enhancing the colonization of probiotics with functional metagenomics, and the other part was doing promoter engineering using this high-throughput barcode sequencing-based approach. Together, hopefully these 2 methods will allow us to open new routes to treat disease using living therapeutic delivery vehicles, and will allow the activity of these vehicles to be precisely controlled to your therapeutic window of interest. Before I end, I want to thank the people who I worked with, not only directly on these projects, but also in the labs that I worked in particular the Dantas lab shown here. Is composed of wonderful people who are great to talk to and do science with, as well as the funding agencies, the NIH for funding myself, as well as the project that I worked on. I want to give a quick plug for my future directions. Coming January 2018, I'm going to be starting my independent lab at NC State University. So if you are interested in collaboration or just simply chatting about that, I'm happy to talk about that as well. So thank you very much for your attention, and I'll turn it over for questions now. Thank you.
Thank you, Dr. Nathan Crook, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, in your colonization experiment, did you observe or look for any horizontal gene transfer occurring?
Thank you for that question. It's a great question, and we did not look for it during our experiments, but it's something very important because you can imagine that improving the colonization potential of a probiotic may, at some point in the future, allow transfer of that genetic material to a pathogen, and we wanted to avoid that. So what we made sure to be careful of was whether the annotation of our recovered gene fragments was a known virulence factor for a pathogen. We did not want to confer virulence to our probiotic. But it's a fascinating question that it should be looked for in future experiments, and you can envision different ways to reduce horizontal gene transfer, given that this through different engineering approaches as well. But thank you very much for that question.
Thank you. Our next question is, in your colonization experiment, did you look for genomic evolution of your host strain as your selection proceeded?
Thank you again for that. It's a great question, and I think genomic evolution is something that is critical to take into account. We tried to take it into account a little bit during our second experiment, where we retransformed these strains into a new fresh genetic background. But genomic rearrangements would be very exciting to look into, and we're currently looking into it right now, where we take the strains that we recovered during selection and doing whole genome sequencing to identify whether there are E. coli miscyl genes which are either inactivated or actually engineered by selection to improve colonization. So that's a great question and something that we're actively looking into.
Thank you. It looks like we have time for one more question. You mentioned the use of unique molecular identifiers in your promoter engineering project. Did you test to see whether including those improved the accuracy of your method?
So that's a great question. Unique molecular identifiers are something that has been used a lot in the immune repertoire sequencing field, and it hasn't gained a lot of traction in the microbiome yet. So I wanted to test it out to see if it worked. What we saw when I analyzed my data was that the unique molecular identifiers, whether I counted the number of promoter-specific barcodes themselves or the number of unique molecular identifiers, I got a very similar result. So it indicates that at least in my control experiments, they didn't matter a whole lot. I'm excited to retest that sort of idea in my in vitro and in vivo experiments, given that there might be reduced abundances of different DNA species and also maybe more PCR cycles involved, which may expand the sort of PCR bias you get. So I guess my answer to that question is hold your thought, and maybe it'll be relevant in the future.
I would like to once again thank Dr. Nathan Crook for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.